All right, so this lecture covers the objectives, or objectives for chapters 2 and 3. So I've played with the organization of this, and I go back and forth. So just bear with me. It will cover everything eventually. I know a lot of my online lectures are long, but I kind of get on a roll and go for it. So remember, you can always pause these. You can speed up my talking. You can come back to them, rewind them, however you want. So don't fear a long video, because remember, you are in control and you can watch it in chunks. All right, so we're going to talk about bonds, a little bit about membranes, about the macromolecules. Again, hopefully most of this is review from your General Biology 1 course. So just to get you thinking back about molecules, remember we have molecules are made of atoms. So you might have a carbon atom or a hydrogen atom or an oxygen atom. And all of these atoms are made up of 3 subatomic particles, which are your, out here, electrons. E minus because they have a negative charge. And in here, your protons and neutrons. And maybe they're labeled opposite, I don't know. Doesn't matter for this. Okay? So what I want you to remember, and protons have a positive charge and neutrons have no charge. What's important to remember is that the electrons orbit around the protons and the neutrons. So they're constantly moving. And the electrons are what are available, the subatomic particles that are available for forming bonds. So this is how we connect atoms to make molecules. So I want to review for you the different types of bonds, because we will talk about them and assume that you know what these words mean. This is just a diagram, again, to remind you that it's the electron interactions that help form bonds. We're going to talk about 3 types of bonds. We're going to talk about covalent bonds, which means you're sharing electrons. So these are 2 atoms, atom 1, atom 2, that have come together and they share the electrons, and it holds the atoms together to make a molecule. This is showing a covalent bond here. This is showing more of how electrons are shared, so an electron from hydrogen and an electron from carbon. We're also going to talk about ionic bonds. And ionic bonds are different in that these are based on charge attraction. We'll talk more about these in a minute. We're not going to be drawing molecules in this class, but I want you again to understand why these bonds are important and what the different ones mean. The last one that's not on here that we'll talk about are hydrogen bonds. These are the weakest bonds, but totally essential for biology and life, and they are due to partial charge attraction. And if you feel like watching a really cute review of bonds, I love Dogs Teaching Chemistry. You can watch this couple-minute YouTube video. Okay, so let's also write here. Covalent are the strongest bonds. Hydrogen bonds are the weakest. All right, so let's look at covalent bonds. And we're going to talk about two different types. We're going to talk about polar and we're going to talk about nonpolar. Nonpolar means there is equal sharing of the electrons. This also means that there's no charge, no full or partial charge to the molecule, or that part of the molecule with covalent bonds. Okay. These are really important bonds. For instance, when you hook amino acids together to form a protein, right, plus amino acid to form a protein, this is formed by covalent bonds. It means they're really strong bonds. They're going to hold that protein together. So covalent bonds hold our monomers together, and we'll start talking about that in a minute. Nonpolar means, or another word is hydrophobic. These types of bonds don't really like water because there's no charge. And so I'm going to be using hydrophobic, nonpolar, polar, hydrophilic over and over for the next few minutes so that you can get a good feel for the different types of molecules, because this is going to be important in our next unit when we talk about things crossing the membrane and how membranes are formed. Okay, so you can have a covalent bond. So we would, I should have put the other diagram here. We would draw these as nice little lines between, oops, the carbon and hydrogen atoms. Nice strong bond. You can have double covalent bonds. Okay, so this is oxygen or carbon dioxide. Okay, again, double strong bonds. They're non-polar. And this is one of the reasons that oxygen and carbon dioxide can cross membranes, because they're nonpolar. They don't have any charge to them, and they're really small. Another place in biology where double covalent bonds are really important is in fatty acids. Okay. So we will come back to this a few more times this semester, but saturated fatty acids have all carbon-carbon bonds, and they have lots of hydrogens. You see all these hydrogens. And this part right here is non-polar. Okay. Fats, lipids, don't like water. And the reason they don't like water is because there's no charge there. There's no way for them to interact with water. An unsaturated fatty acid means it has at least one carbon-carbon double bond. And unsaturated means it's lost some hydrogens. So if you counted up the hydrogens, it's actually missing two right here. So it's not saturated. It doesn't have the maximum number of hydrogens, and that's because it has carbon-carbon double bonds. Okay, cool. So what? The so what is, when we talk about membranes, we're going to talk about how saturated and unsaturated fatty acid tails affect membrane fluidity, so how things move in the membrane. And unsaturated fatty acids, I want you to see that wherever you have these double bonds, it takes the tail from being straight, so it's no longer like this, and it has kinks. So it kinks in the tail. So this is called a fatty acid tail. These are due to the carbon-carbon double bonds, and this affects the, again, the fluidity of membranes. So we'll come back to that. So keep these in mind. You will be drawing saturated and unsaturated phospholipids. Another type of covalent bond is polar covalent, and it's polar because there is a partial charge, and the partial charge is due to uneven sharing of the electrons. Okay. So when you have electrons that have what we call different electronegativity, I will show you a chart in just a second. When you have different electronegativity, sometimes a atom holds on to the electrons a little bit tighter. So what we're having here is we're forming water. So this is hydrogen and oxygen has a higher electronegativity. So as these electrons, which are still being shared, circulate around oxygen and hydrogen, okay, they're going to stay around oxygen a little bit more because it has a stronger pull. And that makes this symbol that represents a partial charge. Okay. So partial charges are not full. We're going to talk about full charges with ions in just a minute. But these partial positive and partial negative charges allow molecules like water to form something called hydrogen bonds. And we'll talk about that in just a minute. So whatever atom can hold on to the electrons the longest, since electrons have a negative charge, that part of the molecule will have a partial negative charge, and the other part will have a partial positive charge. Okay, and again, these are going to be super important for hydrogen bonds. So let's talk about this concept of electronegativity. Electronegativity is just a value of electron attraction to that atomic nucleus. So the greater the number, like over here, the stronger or the tighter that atom is going to hold on to the electrons. If you look at this chart, a difference in electronegativity that's less than 0.4, like you will see between a C and a H. So carbon has electronegativity of 2.5, hydrogen has electronegativity up here of 2.1. The difference we write by delta is 0.4. That is a regular old covalent bond, nonpolar. When you increase that difference of electronegativity, let's show between oxygen and hydrogen, right? It has an electronegativity difference of 1.4. That means you're going to have a polar covalent bond. And when you have a really strong difference in electronegativity, greater than 1.8, then actually an electron will be stolen and moved to a different atom, and one atom will have an extra electron, one atom will have lost an electron, and that's how we form ionic bonds. Okay, so we'll look at those in just a minute. So why do we and this is just another schematic of showing you how electronegativity affects bond formation. Right? So if they're sharing pretty equally, you get a non-polar covalent. As they share things unequally, you get the partial charges, and when you have a great difference in electronegativity, you get full charge difference. These are hydrophilic. Anything with a partial charge or a full charge means it loves water. which really means it dissolves in water. When you have something that's nonpolar, it's called hydrophobic, which literally means water fearing. Okay, so let's look at ionic bonds and then we'll go to hydrogen. So ionic bonds can form between atoms with a great big difference in two atoms' electronegativity values. Okay, so if we go back, we've got sodium, where is sodium? Excuse me, with electronegativity of 0.9, and chlorine with electronegativity of 3.0. So this is 0.9, this is 3.0. Their difference is 2.1. And if we look back on our chart, this is going to form an ionic bond, okay? Anything greater than that 0.818 or 2. So that means that chlorine has such a high electronegativity value that it actually steals, electron transfer, steals an electron. So you can see sodium's purple electron is going over there to chlorine. Chlorine now has an extra electron, and so it has a -1 charge. And we write it as Cl-. Sodium has lost an electron, and so now it's a little more positive. It has an A+, +1 charge. And opposites attract, so you get the plus and the minus, and you have this electrostatic or charge interaction that makes the molecule sodium chloride, which is salt. So an ion is going to have a full charge. It could be +1, it could be -1, it could be +2, all these variations, but it's a full number versus what we're going to talk about in a second as hydrogen bonds. Okay, so we've talked about covalent, polar covalent, ionic, and hydrogen bonds, super important to biology. It's also due to differences in electronegativity, but hydrogen bonds are between molecules in this case. So in this case, this molecule is water. And we have another molecule of water. And we have a third molecule of water. And I showed you that these are the lines, polar covalent bonds. And hydrogens in a polar covalent molecule have a slight positive charge, and oxygens have a slight negative charge. What that means for us is that these molecules can be attracted to each other through these partial charges. So a hydrogen bond is between a partial what am I doing? Partial positive charge on hydrogen and a partial negative charge on usually oxygen or nitrogen. I'll show you that in a minute. These are all polar molecules, so they are hydrophilic, like water, and this allows water to have all the unique properties such as being a universal solvent and high specific heat values and evaporative values and all of those good things you probably learned about in general biology. What we're most concerned about is these hydrogen bonds, which are weak bonds, right? So they can be broken. So we know if you heat up water, you will eventually break these hydrogen bonds and these water molecules will float off into the air. But the more hydrogen bonds you have, the more stability you have. So you also know that water holds together in droplets. That's due to the hydrogen bonds. Hydrogen bonds are essential for life. So this is DNA, and all these little dots in here are hydrogen bonds. Okay. This is how we hold together the two strands of DNA between the nucleotides. Lots and lots of hydrogen bonds. They're weak enough that they can be broken for DNA replication and transcription, but they're strong enough to keep the double helix as a stable structure. Hydrogen bonds are also important in RNA structure. So hopefully you remember that RNA is single-stranded, but it can fold. Okay? So you probably are most familiar with this structure, a tRNA, transfer RNA used in translation. And this is a single molecule of RNA. It has a 5' end, and if you follow it all the way around, it's one long strand to its 3' end. But it folds, it interacts, and you can see all these dots. Those are all the hydrogen bonds. tRNA has to have its structure to function. This is one of my favorite images. This is ribosomal RNA, so part of the ribosome that's important in translation. And this is one long molecule. So here is the 5' end, and here is the 3' end. Okay. And if you trace this around, which I'm not going to do because I'll mess up, it's one long molecule, but it folds, and all of these little lines you see in here are hydrogen bonds. So, super, super important in DNA and RNA, so nucleic acids. Hydrogen bonds are also super important in proteins. And we'll talk about this more in a minute. IMF stands for intramolecular forces. And this purple string is representing the covalent bonds of the peptide chain that hold the amino acids together. So it's amino acid to amino acid to amino acid. And what's shown here, we'll talk about amino acids in a minute, are the R groups. But what I want you to understand is these bonds we've been talking about are also important in forming structures such as proteins. So you'll have hydrogen bonds helping a protein fold into its structure. You can have a nice strong covalent bond. You can have ionic bonds. And then you can have other types of interactions we'll talk about in just a few minutes. So one of the things besides understanding the different definitions and characteristics of bonds, or I should say, the definitions and characteristics of different bonds, covalent, polar covalent, ionic, and hydrogen, is I also want you to be able to identify a polar molecule. So that means something that is going to dissolve in water. The most important polar covalent bonds for biology are between hydrogen and oxygen. I'm going to draw it like this with a little arrow, so you know that oxygen is going to have the slight negative and hydrogen is going to have the slight positive. hydrogen and nitrogen. These are forming the hydrogen bonds between the DNA base pairs. Carbon and nitrogen can form polar covalent bonds, as well as carbon and oxygen. So when you see oxygen and nitrogen bound to hydrogen or carbon, you're going to know that these are polar covalent bonds, which makes that part of the molecule polar. And it all has to do back with electronegativity and the difference between the electronegativity values of the different ions. So let's go back to the macromolecules of the cell, such as sucrose. Sucrose is an example of a carbohydrate or sugar. And what I want you to notice is look at all these OH groups. All of those OH groups make this molecule polar. There are slight negative and positive charges all over. So carbohydrates dissolve in water. And you know that because you can put sugar in water and stir it up and the little sugar crystals disappear. This you may or may not recognize, but this is a polypeptide. So that means there are 1, 2, 3, 4 amino acids shown here. And look, again, we've got OH groups, we've got NH groups. So we have lots of polar regions on proteins. Proteins also dissolve in water. And then down here, this is ATP, which is an RNA. No, this one's deoxy ATP, so it's a DNA nucleotide. So nucleic acids. Here's NH2. Here's something that's going to be able to help form hydrogen bonds between the base pairs. It's got a sugar, so it's polar. It's got these charges on phosphates, so nucleic acids are also polar. So carbohydrates, proteins, and nucleic acids are all polar molecules. Hopefully you've realized that I haven't put lipids in. Those are non-polar. They don't have the carbon, nitrogen, carbon, oxygen, hydrogen, oxygen, hydrogen, nitrogen types of bonds very much. Amphipathic molecules mean they are both polar. Let's see, I'm going to write have polar and non-polar regions. So they're a little bit of both. You can see here we talk about something having a hydrophilic head. This is the polar part. And a hydrophobic tail. This is the non-polar part. And what I want you to see is that non-polar does not like water. It moves away from water because it's hydrophobic. The hydrophilic, or the polar heads, like. Aqueous means water. This is something called a micelle. If you throw a bunch of phospholipids into some water, they'll immediately self-aggregate into shielding the hydrophobic tails away from the water and allowing the hydrophilic heads to interact with the water. This makes it most stable. What we'll be talking a lot about are phospholipids. Phospholipids we draw as a polar head or a hydrophilic head and 2 hydrophobic tails. You will be drawing, like I said, a bunch of phospholipids and membranes for your first homework. So you can always refer back to these images. So this is an amphipathic molecule. Other amphipathic molecules that are really important in our life are detergents. And detergents have a hydrophilic region. You can see there's a charge here, so it likes water, and a hydrophobic region, which is no charge, nonpolar. And so the way that you get that grease off your hands, if you kept washing with water, right, all the grease is going to stay away from the water. It doesn't like it, can't interact with it. So you need to put in an amphipathic molecule, like a detergent, where it can interact with the grease, and it can interact with the water you're using to wash your hands, and then it can help remove that grease or that lipid off of your skin, in this case. So, amphipathic molecules help make membranes. So we already just talked about a phospholipid. It's got a polar head and 2 nonpolar tails. And when they come together, they form something called a bilayer. Bi in biology means two layers. So a cell membrane has 2 sets of phospholipids. The tails make up a hydrophobic interior. And then we have the 2 hydrophilic sides that interact with water. So membranes, or lipids are really good ways to make a barrier. That's all a membrane really is it's a barrier to keep stuff that is dissolved in water on one side and other stuff dissolved in water on another side. And as we talk about membranes and transport, you'll see that this helps control what can go in and out of a cell, or what can go in and out of an organelle. Amphipathic molecules also make something called liposomes. Liposomes, you can see, is a phospholipid bilayer. And they're really great at simulating a membrane. So we can make artificial membranes out of liposomes. What there really are is a small sac that encloses water and hydrophilic molecules. What's beyond the scope of this class, but something kind of cool, is that liposomes have become a way to deliver drugs or genetic material to our cells inside our body. One thing I want you to remember, and we'll talk about, is that membranes are membranes. Okay, they're all made up of these phospholipid bilayers, and so membranes can interact with each other. And so when we talk about the endomembrane system and we talk about vesicles and transport, this is all based on this idea that these phospholipid bilayers can interact with each other because they're made up of similar components. So once you understand what a liposome is, you actually don't need it technically for this unit, this chapter quiz or unit exam, but it's going to come up in the next unit, so I thought I'd get you thinking about it right now. All of these details you don't need to know. I just like to include things to keep you thinking, and things you might see in upper division classes. All right, so we've been talking about membranes, and we've been talking about cell membranes and organelle membranes as phospholipid bilayers. And I want you to be able to understand the difference between these two terms, semi-permeable and selective permeable. All membranes are semi-permeable. Semi-permeable means that certain molecules can cross a membrane on their own. And we're going to really talk about this in chapter 7 and 8, but again, I want to introduce these concepts to you. Semi-permeable means you don't have any control, okay? So whatever fits, whatever fits or has the right charge, open with a lack of charge, can cross the membrane on its own. Okay. So you don't have control of what goes through a semi-permeable membrane. Selective permeable is where our cells get their functions, and that means we have control, or the cell has control of what crosses the membrane. And this is the whole focus of chapter 8, and it's all based on transport proteins. You're going to have like little gates that can help control what crosses a membrane. In each cell, different types of cells will have different proteins in them, and they'll be selectively permeable to different molecules. All phospholipid bilayers will be semi-permeable to the same stuff because that's just based on the structure of the membrane. Okay, so we've talked about bonds and how molecules are formed and the different kinds of bonds and how they are important in biology. And we've talked about molecules that have both polar and nonpolar components, which make them amphipathic, which led us to membranes and the definition of semi-permeable and selective permeable. Okay, I'm going to flip back now to the macromolecules of the cell. This is a combination of chapter 2 and 3. Our four, I should call them macro, the big molecules of the cell are carbohydrates, proteins, nucleic acids, and lipids. So let's talk about each of these a little bit more. I mentioned earlier that, for instance, proteins are made out of a monomer called amino acids. And so this chart is showing you that proteins, nucleic acids, and carbohydrates are all made up of monomers. And they make, the monomers are hooked together by these different covalent bonds. And we'll talk more about some of the functions. What I want you to get from this slide is that lipids are not made up of true monomers and polymers. So I'm never going to ask you what is the monomer of a lipid, what is the polymer of a lipid, because they don't have nice repeating units. Okay? So for instance, DNA is made up of nucleotides, and when you hook nucleotides to nucleotides to nucleotides, you get DNA, or you get RNA. Right? Proteins, like I said, you hook amino acid, amino acid, amino acid, you get a protein. That doesn't work with lipids because lipids are very diverse in their structure. So here's a few examples of lipids. So you might have heard of testosterone and estrogen. These are hormones. So right now I'm giving you some functions as well as some characteristics. Okay, cholesterol is a lipid. Now, cholesterol and these hormones look very similar, but they look quite different from a fatty acid. And so we've talked about phospholipids that are down here, which is the head and two tails, and this is the head and tail 1 and tail 2. Triglycerides are probably what you learned as the classic monomer polymer of lipids. You have a glycerol backbone and 3 fatty acid tails. 1, 2, 3. That's where the triglyceride comes in. But you can see that you cannot use these same building blocks to make cholesterol or estrogen. Or a free fatty acid doesn't have a glycerol backbone. So lipids really do not fit the whole monomer polymer. What are all these lipids for? So we've talked about hormones. Hormones are important signaling molecules. Triglycerides and other types of lipids are great for energy storage. Many of us have a little too much lipid energy storage. Phospholipids and cholesterol are really important for making membranes. These are hydrophobic molecules. Yes, there's an OH here. So estrogen has a slight charge right at the end, and testosterone. And phospholipids, we talked about amphipathic. But they're mostly hydrophobic, they're mostly water-fearing because of these long carbon-carbon chains. So there's lots of carbons and hydrogens in lipids and very few oxygen molecules. So in general, lipids are hydrophobic. Waxes, if you've ever felt a plant leaf, right? Plant leaves can repel water because they have a waxy coating. That's all lipids. Pigments. If you go review photosynthesis and look at the structure of chlorophyll, that's based on a lipid. It's a big carbon-hydrogen ring molecule. So we've already talked about this. Yes, it's a little repetitive. Repetition is great for learning. So lipids make up our membranes. We're going to be talking a lot about cholesterol in our membranes. We'll be talking a lot about saturated and unsaturated fatty acids in our membranes and how they work in membrane fluidity. You will also get into a little bit more about different types of phospholipids. So there's something called sphingolipids, which you probably haven't really heard of. They're a minor lipid in most membranes, but they're also unique to eukaryotic cells. So we'll talk a little bit about these different types of phospholipids that make up our cell membranes. All right, let's move to carbohydrates. So carbohydrates, a lot of people also call them sugars, has a general C6H12O6 component. I think this is glucose. I'm not going to ask you to identify any of these, but I put this up here to remind you of some of the functions of carbohydrates. And the main one you probably think about carbohydrates is energy. Right? Eat some sugar, little kids get hyped up, you got lots of energy. We also have energy storage in the form of carbohydrates. So starch, things like our breads and potatoes, have lots of repeating units. So the monomer, I almost forgot, is called a saccharide. And then you can have these polysaccharides shown on the left here, such as starch, glycogen, a bunch of sugars, a bunch of carbohydrates hooked together. Glycogen is your storage in animal cells. Starch is the storage of carbohydrates in plant cells. Carbohydrates are also important for structure. So, for instance, cellulose is made up of a bunch of carbohydrates, monomers hooked together, and cellulose makes up plant cell walls, so it allows plants to be rigid. Chitin is another type of carbohydrate polymer that's important for structure, so it makes like the hard shells of crabs. In addition, carbohydrates are really important for cell signaling or cell communication. So if you can read these little words, they say glycoprotein, glycolipid. And what that means is that they have sugars attached to a protein or attached to a lipid molecule. Okay. Glyco means sugar and what it's attached to. So these are important for, on the outside of cells, for cells to be able to communicate with each other. So there you've got some functions of carbohydrates and the general structure. Let's move on to amino acids. So an amino acid, the general structure that I write is like this. We've got a central carbon, we've got an amino group, we've got an acid group, and we always have a hydrogen. And then the fourth part, the R group of an amino acid, which you see here, R1, R2, that's the unique part, and I'll show you that in a minute. But I want you to remember that amino acids are the monomers, proteins are the polymers. They're hooked together by peptide bonds, a nice covalent bond. And when you hook a whole bunch of them together, amino acid, so each little colored ball stands for an amino acid, you get a polypeptide chain, also called a protein. So what are some of the functions of proteins? Well, enzymes are probably the one you're most familiar with. Enzymes are making chemical reactions happen. Proteins are really important for membrane transport. So moving things across the membrane, part of that selective permeability. There are some proteins that are hormones. So there are lipid hormones, there are protein hormones such as insulin, important in communication in the body. Proteins can be important for structure, such as the cytoskeleton in your cell. So proteins have probably the most diverse functions. I've only listed a few here. They're made up, again, of repeating units of amino acids, and I said that each amino acid has an R group that makes it unique. There is a chart in your book, figure 3-2, I believe, that looks similar to this. And of course, you don't have to memorize any of this, but you have to be able to use the chart. And so what I mean by use the chart is I want you to understand that these different R groups, which are all in the colored blocks here, give a protein properties. Because it helps the protein, or it directs the protein folding. And this chart has the basic 20 amino acids that make up most of our proteins. And it's classified into R groups that are nonpolar. Okay, you've heard the word nonpolar a whole bunch of times, so hopefully you immediately think hydrophobic, wants to get away from the water. We have polar amino acids. So you see an OH group on these. And we haven't talked about serine, but a SH group, NH groups, things like that make them polar. We have some amino acids that have full charges. Okay, so aspartic acid has a negative charge here. Lysine has a positive charge. So these are both going to interact with water. The nonpolar amino acids are going to want to get away from water. And where that comes into play is as a protein folds. So there are 4 levels of protein structure: primary, secondary, tertiary, and quaternary. The primary structure of a protein is just the sequence or the order of amino acids. So the order, the number of amino acids you have, if you look back here, this strings on the bead is all primary structure. What happens for secondary structure, okay, so primary structure is all made by covalent bonds. Secondary structure is made by hydrogen bonds. So hopefully you see the little dot, dot, dots, right? And secondary structures are repetitive. They're made by the interaction between the carbon end of one amino acid and the amino end, I should say the carboxyl end, of another amino acid. So they're based on the backbone, this sequence. You can have secondary structure that's called an alpha helix. That's what's shown right here. Or you can have beta pleated sheets, which are more flat, and I'll show you some examples in just a minute. Okay, so secondary structure is based on hydrogen bonds and the interaction between the backbone, the hydrogens, sorry, that's not what I wanted to do, the carboxyl group and the amino group. Does this look so weird? Oh, because it doesn't have the full thing. So yeah, so these two could interact. Sorry, tertiary structure is where the R groups come in. Tertiary structure is based on R groups and our intramolecular forces. So back to this diagram we talked about, if this is our primary structure, our sequence of amino acids, where the different R groups interact, those are the intramolecular forces. And something we hadn't talked about yet was the hydrophobic interactions and the van der Waals. So, amino acids that don't like water, that are nonpolar, are going to move to the inside of a protein as it is folded. And the ones that are electrically charged or polar are going to be able to go to the outside of the protein. Last one I don't want to forget, quaternary structure means that two or more proteins interact to form a functional protein. Okay. And sometimes people don't call it proteins, they say two or more polypeptides, because actually the term protein implies function. I'm not going to get picky about that. Know that tertiary structure means two or more of these polypeptides coming together to form a functional unit. This is an example of tertiary and quaternary structure. So first let me point out that these arrows, these flat arrows, represent the beta-pleated sheets, and that's secondary structure. And the curly Q's represent the alpha helices, and that's also secondary structure, right? But this whole crazy folding is called tertiary. So inside here are the hydrophobic amino acids, and along the outside are the hydrophilic amino acids. Now, when you combine more than two or more, I should say, polypeptides, you get quaternary structure. So this is 1, the yellow is 2, the green is 3. 3 non-identical amino acid chains will make up a G protein. So individually they're not functional, but when they come together they form a functional unit, and that's quaternary structure. The other thing I want you to know is that these amazing structures are spontaneously folded. So proteins have self-assembly, which means that all these different types of intermolecular forces and bonds push the protein into this most stable structure. So as a protein is translated, it will start interacting and become the most stable structure it can. You can imagine if you have a mutation somewhere that changes an amino acid, you can disrupt this protein structure and disrupt the function of that protein. And you probably, if you haven't taken genetics yet, you will talk about that in genetics, or if you have taken genetics, hopefully you remember that. Okay, so that covers protein structure. The last slide I have for you is this concept of directionality of nucleic acids and proteins. Hopefully you remember that DNA and RNA have 5' and 3' ends. That's its directionality. We usually write it 5' to 3'. This tells us about how the polymers are formed. The directionality for proteins is that you have an N-terminus and a C-terminus. So this is your NH2 or NH3+, This is your COOH or COO minus. Same difference. And proteins are always made from the N to the C terminus. So the new amino acids are always added to the C terminus, just like the new nucleotides on a DNA or RNA strand are always added to the 3' end. So again, a lot of this is just reminding you, getting you caught up on terminology that will be used throughout the semester. So I hope this helps you with the chapter 2 and 3 learning outcomes or learning objectives. And that's it.